All right, hi everybody. Today I'm going to make you all believers. So here we go. One of my favorite questions to ask my students, do you believe in evolution or not? And I'd say that quite often I get 30 to 40% of my students say no, they don't. And then a follow-up question I usually ask is why or why not? So a follow-up question I like to ask is, what is evolution? And is it important to know about something before developing an opinion on it or not? Now, this occurs later on in the semester after I've covered it, and they're usually able to give me a pretty good definition then, but interestingly, they still state that their opinion really hasn't changed, although they agree it's important to understand what you're talking about. So they don't connect those two. So when I ask why, or if I ask about evolution in general, and whether they say yes or no early on in the semester, they generally, my students will give me a wide range of answers as to why they say no. And some of those include, it's just a theory, there's no proof, doesn't make any logical sense time-wise, nobody's ever seen it happen, and often there's wide ranges of religious views that interfere with that. However, I'll say even with many students that say, yes, they believe in evolution, even their evidence or their examples are really still not quite correct, meaning that it seems like whether they believe in evolution or not, they still have trouble early on really defining it. So that makes sense to me, and that's what we're going to talk about today, what evolution is, what it's not, and we're going to clear that up. Now, when most people think of evolution, they think of this guy, and that's really not incorrect. Charles Darwin is generally credited with the theory of evolution by natural selection, although others were involved with it. Darwin's work relied heavily on examples from artificial selection, which I'll discuss later. And you can also check out my Darwin video, and I'll put the link below for more on Darwin there. So the real reason why evolution is such a powerful and strong theory, and I'll go into that later on too, in biology today is because of a movement called the modern synthesis that really took place in the 20th century, in the 1940s. And that allowed evolutionary biologists, geneticists, and developmental biologists to sort of come together and reconstruct evolution. And this is how we use it now. So Julian Huxley, evolution, the modern synthesis, sort of got that ball moving. It's continued on that path since then. And it's quite different than what I think people are used to thinking when they think about Darwin. So we'll discuss why that is and why that's important. We need to start off with some definitions for evolution so it makes sense, and then we can realize how scientists often refer to evolution in two different extremes, and we call those micro and macroevolution. And we'll start with this, the idea that when we talk about evolution, we're talking about genetic change in a group population of organisms over some period of time. And we can break those up into micro and macroevolution. Today I'm going to focus on microevolution. But the idea is basically this. In microevolution, the idea is we have relatively small genetic changes in a group of organisms, a population, over a relatively short period of time, whereas in macroevolution, we have relatively large genetic changes occurring in organisms over a relatively longer period of time. So same sort of mechanisms, but it's the idea that you can accumulate larger number of changes over time. So they're like two bookends, really. It's not necessarily a black or white. Now, I want to say that when I started this video and I talked about I'm going to make you all believers when I was done, the real trick is I'm not going to prove that your personal or religious views are wrong. I think probably most people do not understand what microevolution means and how we use it and how we can measure it, and that's where the strength of what I'm going to say comes in today. So let's start with a hypothetical example, and we'll talk about the microevolution of a made-up group of rabbits as a hypothetical example, and then I'll show you some real examples that people have actually studied. So let's say you have a population of rabbits, and like always, there's usually some kind of variation. So let's say some of the rabbits have white fur, and some have black fur and brown fur, and some are tall, and some are long, and some are bigger than others, and some have long ears and so forth. And we have this sort of variation, which we find in most things. So we have some kind of genetic variation. And then imagine our initial population looks something like this, where I have a whole bunch of variation in these rabbits. And then for whatever reason, let's say there's some type of predator wanders into our rabbit population, and it just happens to find and eat the lightest color rabbits. Maybe the white rabbits are easier to see, but whatever reason it is, our predator comes in and it wipes out the white colored rabbits, or it finds those easier to get. So rabbits can have, it only takes a few months for a rabbit to go from being born to being able to reproduce. So really in a year or two, you can have 2 or 3 generations of rabbits, and you can have anywhere from like 5 or 6 rabbits to 10 to 12 rabbits. So it doesn't take long within a year with rabbits. That's why I pick it. Also, it's one of the only things I can draw even, a little bit. If I'm not very good at drawing. But anyway, so we have the same thing. Our predator comes in, eats those. And so over time, what we see by our 3rd generation, we end up with this very different looking population. Maybe we still have a couple of the light ones for whatever reason, and you can get into the genetics on that, but we end up with, maybe within just 1 year, starting with a certain population of rabbits and ending up with a different number of those looking quite different at the end. So if we started with 6 rabbits and all the white ones got eaten, or most of them did, would we be able to say if evolution occurred or not? And so the answer is, if color variation in these rabbits is genetically determined, not something they ate or that kind of thing, and if over time the relative frequencies of the genes that were present for light and dark colored rabbits and so forth are different, they've changed, then yes, evolution has occurred, because evolution is defined as that genetic change in a population over time. So nothing new has to arise from that, although that's what a lot of people are wanting with their definition of evolution. You just need a change in the frequencies of certain genes or alleles over time to show that evolution's occurring. Let's talk about some real examples now. Bacteria make some of the best examples, and I'll just go way back to a famous study, Joshua and Esther Lederberg, University of Wisconsin, in 1958. Their now famous experiment, which has been replicated over and over again in all kinds of different ways, and they have a nice example of antibiotic resistance that they're famous for studying, won the Nobel Prize for it. And so they start with some bacteria that are susceptible to the antibiotic called streptomycin. And what that means is that if the bacteria are exposed to the streptomycin, then they're not going to be able to survive and reproduce and all the things that bacteria does. So this would be good, by the way, if you were infected with this kind of bacteria, in the sense that you could then take streptomycin as the medication prescribed by your doctor, and you could wipe out that infection with it. Okay? Now, what Joshua and Esther Lederberg discovered within a short period of time, only takes, several days or weeks before, and it's sort of random that they're going to acquire this mutation, because as they copy, as we'll see later on, as you make copies of this, you make a mistake, and you can then pass on that mistake to other future generations. But a mutation can occur and spread through the population very, very quickly, and this happens such that we can get evolution in these bacteria since 1958, and it's been well studied since then, in just a few days, that are now resistant to streptomycin. And since then, we've had dozens and dozens and probably hundreds of studies on antibiotic resistant strains of bacteria that are changing over time, becoming more resistant to our antibiotics. One of the best studied evolutionary mechanisms probably that we use now So when we use the word theory in biology, what we mean by that is that multiple studies from quite often from very different areas have kind of all concluded on some general principle that we see over and over again. So the theory of evolution by natural selection is called that because so many studies in so many different directions kind of solidify the fact that this is a mechanism that we can count on. All right. And another example, Peter and Rosemary Grant working on Galapagos finches. That's right. It's the same finches Darwin studied, and people have continued to research them, including Peter and Rosemary Grant, who've been studying them for 40 or 50 years now and have graduate students that go on and study them. I believe they're at Yale University or Princeton, Yale or Princeton. It's one of those, two. I should know that. They'd be upset probably by that. But anyway, and so what they found in one of their studies is that we have this particular, I think this is a large ground finch. It's one of the Darwin finches. There's a whole bunch of them. And what they found as they have been sort of measuring really everything on these birds over time. And so this is really sort of, you get a little bit lucky. So it's not like they set out to do this, but rather what they did was they've been studying these finches for a long period of time. And one of the things they found was that in the 1970s, around 1976 or so, when they measured all these birds, there was this sort of average bill or beak depth, right? Certain size to the beak. And then what happened was a drought hit the island shortly after that. And what they found was that in that short period of time, in just a couple of years, that drought, which wiped out a lot of the plants with sort of the thinner seeds coating them, they found that the ground finch ended up with this slightly large, the average bill shifted. There was this population shift where what was favored was instead this slightly larger bill. So that's a good example there where nature does some kind of thing, creates a selection force, and we actually can measure it and see that in these finches, even over a short period of time. Okay. Another great example, these Anole lizards found on the Caribbean islands, and there's a whole bunch of Caribbean islands, there's a whole bunch of lizards on them. And in this particular one, what they found is hurricanes hit this area quite often, and Colin Donahue and his research group found that areas hit by these hurricanes had these really quick evolution in toe pads. And as you can see in the photo, the hurricanes require these lizards to sort of hold on to the branches of these trees, and probably some don't do so well. And so you can see they're holding on, and if they don't hold on, they get sort of blown off. And there are all kinds of, I'll put some links down below. There's thousands of peer-reviewed journals from Microevolution at this point. So now we're going to talk about some basic microevolution mechanisms. We have selection, which we'll go into a little bit, genetic drift, gene flow, and mutation. So we'll cover all four of those. Those are our four basic ways that genetic change can occur over time. So first we'll start with selection. And selection can be broken down into two groups, one being natural selection, and the Peter and Rosemary Grant natural selection is an example of that, where you have some kind of force that changes a drought, wind, the lizards on the Caribbean island in that case. Those are examples of natural selection. But we can also have artificial selection, and one of the wonderful things about what Darwin did is he worked on pigeons. He bred pigeons, and he did research on dog breeds. And so a lot of what Darwin did and concluded was actually nicely quantifiable from studies on artificial selection, which we had been doing for, thousands of years, but nobody really sort of put together this mechanism of how it could be done, and that basically nature could be doing the same thing rather than humans deciding. So that would be artificial selection. And either way, whether it's natural selection or artificial selection, we have three sort of forces that can push evolution by natural selection or artificial selection. So you can have what's called stabilizing selection, is whatever's in the middle is generally what's the best and more favored. So maybe the tallest of the animals and the shortest of the animals, they kind of don't do well for whatever reason, and rather it's the middle point. Or you can have directional selection where the smaller something is, the better, or the bigger something is, the better. Or it could be a fur coloration. So maybe it's the darker the fur, the better, or the lighter the fur, the better. Either one of those is what we call directional, because it's pushing the evolutionary force, or artificial force is pushing the selection to the right or left. And then it could be disruptive. Disruptive is where the middle isn't as good, but rather the extremes are sort of selected. So maybe there's an advantage to either being really big or being really small. Maybe if you're really big, you can fight off a predator, but if you're really small, you can hide under a hole. But being in the middle, you're kind of out of luck. Genetic drift occurs when you have some small sample size that ends up later representing your whole group. We'll start with these big animals here. These are called northern elephant seals, and 100 to about 150 years ago, they were hunted to extinction. So prior to that, there were all kinds of color variations. I hunt them all to extinction, and there were, like I said, 20 to 50 or to 100 that survived. They reproduced. When they came back, you lost a lot of the color variation, so they became kind of more uniform, and that's what we see now. Okay. Another example of genetic drift is called the founder effect, and it is similar, and it utilizes, once again, small sample size. And in this case, what happens is we have these, let's just as an example, let's say we have these islands, and on these islands we have a bunch of lizards, and I'm using these picture of an island, and I'm using these lizards, but just in case anyone notices, these islands here are in Australia, these lizards are in the Caribbean. I'm just putting the idea together. So these lizards, I know, do not exist on those particular islands. Let's say I had lizards on one island, and then something happened, one of those hurricanes or something, and it blew some of them to another island. And if on my one island I had brown and green ones, but during the hurricane only a few brown ones or a few green ones blew to the other island, and if that other island had no lizards on it, then the new starting population, founder effect, is with just those starting population of lizards. And so as those reproduce, those end up representing a different combination or percentage of frequency of what I had before on my previous island. The other thing that could happen is what we call gene flow. And gene flow occurs when organisms move from one population to another. And again, that's a good example of a population evolving. So even though you don't get anything new in the world of things, that population is picking up something new. So imagine you had some kind of animal, and it could be, it could be those lizards, it could be frogs, it could be butterflies, it could be anything. And let's say on one island, they tended to be one color and they were different on another island. Well, if a couple of those wandered off from one island to another, then it changes the frequency of the genes on that island, and we call that gene flow. Gene flow is where genes move from one population to another, thereby changing the frequency of that population, thus evolution. Okay. Now, the last one I mentioned already, we talked about bacteria. Nope. Okay, so the last one is our only source really of new genetic information, and we call this mutation. And we mentioned mutation with bacteria a little bit ago, but let's look at the machinery a little bit closer so we can see in a living organism how that might work and how that might affect what we're discussing. So when you take DNA, you take any organism, it doesn't matter if it's a human or a dog or whatever, there is DNA inside that organism, and that's what codes for all the different proteins that make a person or a dog, either a dog or a person, and have, different hair colors and different skin colors and eye color and all that kind of stuff, because the DNA is where the genetic material is, the genes, and that codes for what you get as what we call a phenotype, or the appearance of some organism after that. So the enzyme that's involved in doing this is an enzyme called DNA polymerase, and it goes along and it copies that, but every now and then it makes a mistake. Overall, it's pretty good. It makes a mistake once out of maybe every 100,000 times it copies, and then it has some mechanisms that fix that. So even then, you're only making a mistake maybe one every million base pairs or so, so it's pretty good. But over a period of time, we all end up eventually having some mistakes made in there. So a mutation occurs in a place where the DNA changes, and it can change because DNA polymerase made an error in copying. It can change because of certain chemicals that can affect your DNA. UV radiation is one of them, X-ray machines, smoking. So there's a few chemicals that can alter and damage your DNA, which is why, they're bad for you in part. So what happens is a mutation occurs when you change that DNA. And once you change that DNA is now then different. And that happens every now and then, but like I said, most of the time you fix it. But by the time you're age, 19 or 20, even with the fixing part, you tend to have an increased risk where you've captured or trapped one of these things, or you've made this mistake in a place that is it's not good. In fact, this is one of the sort of leading ways we describe how cancer occurs, because there are genes that regulate cell growth, and by having a mutation in one of those genes, you can alter the ability to regulate that cell growth, which causes trouble. Now, before you freak out and you get worried about that, like I said, the mechanisms to copy the DNA are usually very, very accurate. They make a mistake once every 100,000 times or so, but then there's genetically, but then there's molecular, but then there's parts that fix the mistakes. So that's not so bad. And the mistakes can be really extremely bad. In fact, when we talk about cancer, we're mainly talking about these DNA replication mistakes as being the root cause. Now, before you freak out, some of these mutations are in the third position of what we call a codon, which you'll learn about later on. And this is what we call the Wobble Effect. And what that means is that makes it okay. So if you look, for example, at this chart that shows you all the different genetic codes and for the amino acids they code for, if you look at, say, phenylalanine here, if your mRNA sequence that comes from your DNA is UUU, but you have a genetic mutation that changes it such that your mRNA becomes UUC, what you find out is, although that change is there and a mutation's occurred, you still are putting in the correct amino acid. So functionally, that should still work the same and make everything okay. I should also point out, if you've had genetics before, or I should also point out that what I'm describing here is really called a point mutation, just to sort of let you know about the effects of how a mutation affects evolution. So there are many different kinds of mutations, and those can affect our understanding of microevolution as well. But in this particular introduction to it, I'm just trying to cover the basic idea that mutations affect the genes you have in your population. Okay? Now, the last thing I'll say is that mutations can be good, or they can be bad, or they can be neutral. Now, usually I would say most people with the word bad, they think, okay, I understand a mutation is bad. So what that means is something goes wrong, right? So if you have a mutation and it changes your DNA such that you're more likely to develop cancer or some other catastrophic illness, then that obviously is pretty clearly a bad mutation, or a negative mutation is probably a better way to word it. However, there are neutral mutations like I just described here. If I use leucine, for example, this time is a different example. If I have CUC, but I change my code to CUA, I still get leucine there. That's more like a neutral mutation because really nothing has changed. I've changed the DNA, but the end result is I'm still getting the right amino acid, so everything's okay. But what about a good mutation? How do you get a good mutation? And this is what students, I think, struggle with the most. How can a mutation be good? Well, if the mutation, whatever it is, results in an increase in fitness, then that is what we call a good mutation, or a beneficial mutation. Now, fitness is not working out at the gym better, necessarily more muscle, faster swimming times, faster running. That's what we usually think about as fitness, is your cardiovascular overall personal fitness. What I mean by fitness here is what we use fitness, we use the definition of fitness in ecology and evolutionary biology and animal behavior. Fitness is your ability to survive and reproduce and have babies. So if you have a mutation that increases your ability to live longer and have more babies, or it makes you better at getting food, and therefore because you got more food, you could have more babies, and those babies survive and go on. Any of those kind of things that affect reproductive rate and therefore increase the odds of spreading those genes, that is what we call a beneficial mutation. So in closing, that's it. Microevolution, 4 methods by which change can occur. And hopefully you picked up, as I mentioned before, my goal of this is to hopefully educate you on what the word evolution means. And when I say I'm going to make you all believers, the idea is not that I'm infringing upon anyone's particular religious or personal views, but rather I'm trying to point out that what you've been taught about evolution is probably wrong. And hopefully that clears things up for you. Go tell your friends.